All right, let's just dive right into this. Welcome to today's explainer, where we are essentially going to act as cellular detectives. We're diving into a really fascinating lecture by Professor Sameh A. Hassanayan, and it's all about data science in healthcare, RNA-seq data analysis. Now, if you've ever wondered how hardcore data science intersects with the literal building blocks of life, you are absolutely in for a treat today. We're going to take some incredibly complex, university-level biological and statistical concepts and just break them down. We'll make sure the foundational building blocks are super clear so anyone can follow along.
So let's start with a really big question. How in the world do we know what a microscopic cell is actively doing or thinking at any given moment? This isn't just some fun philosophical thought experiment. It's an absolutely vital, real-world problem. Think about it. If we want to cure diseases, if we want to understand genetics, or develop crazy new targeted therapies, we have to know exactly what a specific cell is up to right now. But we can't just ask it, right? We need a scientific way to read its mind, so to speak.
To solve this mystery, we have a clear roadmap for today. We're going to break this complex topic into 4 digestible steps. First up, reading the cell's messengers. Second, the simple counting trap. Third, normalizing the data. And finally, number 4, beware of batch effects. Okay, jumping right into our first section, reading the cell's messengers and how we actually decode those tiny biological signals. To really understand this, I want you to think of your DNA as a giant master library blueprint. It literally contains the instructions for everything your body will ever need to do. But the cell doesn't just use the original blueprint every single time it builds something. That would be way too risky. Instead, it makes these temporary, disposable photocopies of specific instructions, and those are called RNA. This brings us to a crucial definition from Professor Hassanayan's notes, the transcriptome. The transcriptome is the complete collection of all these RNA transcripts, like messenger RNA or mRNA, inside a cell for a given physiological condition. So if your DNA is the entire library building, the transcriptome is the actual stack of books currently sitting open on your desk. It shows us precisely what the cell is actively working on right now under these exact conditions. So the next logical question is, how do we read that stack of books? Well, we do it through something called RNA-seq. According to our source material, transcriptome sequencing, or RNA-seq, is a high-throughput sequencing technology. It's used to rapidly and comprehensively obtain the transcriptional status of biological samples at a highly specific time. You can kind of think of RNA-seq as our technological magnifying glass, or maybe like a rapid document scanner. It allows us to quickly scan and read all those checked-out books all at once, capturing this perfect frozen snapshot of the cell's activity at that exact split second. That leads us into section 2, which really highlights the core problem of this whole lecture, the simple counting trap. We're going to see exactly why basic math totally fails us here. Now that we have our fancy magnifying glass, you might be thinking, hey, why can't we just count up all these RNA messengers and call it a day? Like, if we see 10 copies of RNA for eye color and only 2 for hair growth, the cell must be prioritizing eyes, right? Well, not quite. Our source text outlines 3 massive hurdles that make simple counting practically impossible. First up is ambiguous mapping. Genes can actually create multiple structural variations of themselves, which are called isoforms. Imagine finding a torn page on your desk that could perfectly fit into 3 different books. Simple counting gives us no mathematical way to know where it truly came from. The second hurdle is sequencing depth variations, which is where the total size of our data pools varies wildly between different samples. And the third is positional and sequence biases. This just means our tools naturally tend to favor reading certain sequences over others, which obviously leads to really uneven and totally inaccurate counting. Let's actually illustrate one of these biological hurdles by looking at a direct question straight from Professor Hassanayan's quiz materials. It asks, when comparing raw read counts within the exact same sample, why might gene X show more reads than gene Y, even if they have the exact same actual transcript abundance? The answer to that mystery is feature length bias. As the text explicitly points out, longer genes naturally produce more reads. Let me give you a concrete example to make this super clear. Imagine 2 people are sitting in a room reading. One is reading a tiny 5-page pamphlet, and the other is reading a massive 500-page novel. If I take a random split-second audio recording of all the words currently being read out loud in that room, I'm going to capture way more words from the novel. Not because the novel is being read more intensely or more often, but simply because it's massive. It takes up more space. In biology, if a gene is physically longer, our sequencer will naturally spit out more reads for it. And that completely tricks our raw data into thinking that gene is way more active than it actually is. The other huge trap is sequencing depth variations. The source notes that library sizes, and by that we mean the total number of reads we extract, vary drastically across different samples. Think of this like trying to count fish to figure out how populated two different lakes are. If you scoop a tiny little sand bucket into Lake A, and then you drag a massive, miles-wide commercial net through Lake B, you obviously can't just compare the raw number of fish you caught to see which lake has more fish per gallon. The tools you used to sample the data were completely different sizes, so we absolutely have to equalize the volume first.
And because of that pamphlet versus novel problem and that tiny bucket versus giant net problem, raw counting is completely useless, which perfectly brings us to our third section, normalizing the data. This is how we level the statistical playing field so we can make true apples-to-apples comparisons.
Our source document highlights a few different mathematical metrics to fix this, specifically comparing things like RPKM, which stands for reads per kilobase million, and TPM, or transcripts per million. Now, without getting completely bogged down in the scary math, here is the absolutely crucial takeaway. These are simply two different statistical methods used to find a proper balanced baseline. According to the lecture, RPKM is great for comparing different genes inside a single sample, like, say, comparing gene X and gene Y in the exact same patient. But TPM is the optimal standard for intersample comparisons, meaning if you want to compare the data from patient A over here to the data from patient B over there, TPM is the metric you need to use to ensure those different library sizes are perfectly balanced. Okay, so we've normalized the math, we've balanced our library sizes, and we're totally done, right? Actually, scratch that. We are not done. Let's move to our fourth and final section, Beware of Batch Effects, which deals with the physical artifacts of the lab itself.
Because as it turns out, even with absolutely perfect, flawless math, the physical environment can still completely lie to us. The lecture notes strongly emphasize that batch effects are widespread in high-throughput biology. They define these as artifacts that are entirely unrelated to the biological variation we actually care about. For instance, imagine you have the exact same recipe for a cake, but you bake one cake on a crazy humid day in Florida, and you bake the other on a bone-dry, freezing day in Alaska. Those cakes are going to come out completely different, even though the core instructions, the recipe, were identical. In our data, experiments processed on 2 different days might show wildly different results just because the room temperature shifted a few degrees, or honestly, even because 2 different lab technicians happened to handle the test tubes a little differently.
And this next part really illustrates just how incredibly deep the problem goes. The source material visualizes this as this complex, tangled web of arrows showing how outside variables infect the data at every single stage. If you look at the top of the workflow, you have things like personnel effects and environmental conditions, and they're pointing arrows all the way down into growing the organism, taking the tissue sampling, doing the RNA processing, straight down to the data extraction. Meanwhile, from the bottom up, things like the specific batches of chemical reagents used or the weird little quirks of a specific machine's equipment shoot arrows right back up into the hybridization process. Every single step of the workflow is vulnerable to the messy physical world altering the data. So, just to summarize these pesky non-biological artifacts, the major sources you always have to watch out for are personnel effects, environmental conditions, batches of reagents, and equipment effects. As the lecture firmly points out, if we aren't incredibly careful, these batch effects can substantially confound downstream meta-analyses. For example, if you're trying to compare a cancer study from a lab in Tokyo to a cancer study from a lab in London, you might think you've discovered a massive new biological breakthrough, when in reality, you've just discovered that the 2 labs bought their chemicals from 2 different suppliers. This all wraps up into a really humbling summary quote directly from Professor Hassanin's slides. He states, RNA-seq normalization is difficult, still an open statistical problem. I love this because it's such a great reminder that finding a proper baseline isn't just a neatly solved equation you find at the back of a textbook. It is an active, ongoing scientific challenge that requires constant vigilance from data scientists. Which leaves us with this final, somewhat provocative thought to ponder today. If our very tools, gene lengths, sequencing depths, and daily physical environments introduce bias at literally every single step of the pipeline, well, how much of our biological data is truly biological? It really highlights the absolute critical importance of robust data normalization in all of modern science. Because without the math to level the playing field, we aren't reading the cell's mind at all, are we? We're just reading the static noise of our own instruments. Thank you so much for joining me on this explainer, and I really hope this helped you decode the wildly fascinating world of RNA-seq data analysis.